Hey everyone, and welcome to The Explainer. Today, we're diving into a really clever chemistry trick that lets us measure something we can't even see. It's a classic problem, but the solution? Well, it's surprisingly visual.
Seriously, think about it for a second. How in the world do you measure something that's totally invisible, just dissolved in a solution? It sounds kind of impossible, right? But chemists have this fantastic technique to do exactly that, and it all comes down to a really dramatic color change. So, how do we pull this off? How do we measure the unseen? Well, the answer is a powerful method called titration. And trust me, this is an absolute cornerstone of chemical analysis.
At its heart, titration is basically a super controlled chemical reaction. So imagine you've got a flask with your mystery substance. In chemistry speak, you call that the analyte. Then you very carefully, drop by drop, add a solution where you know the concentration perfectly. That's your titrant. By keeping track of exactly how much titrant you needed to finish the reaction, you can calculate the amount of your mystery analyte. Pretty neat.
Now, there are a bunch of different types of titrations, but today we're going to zero in on a special kind called redox titration. Redox is just short for reduction oxidation, which basically means the reaction involves electrons moving from one thing to another. And our key player for this is going to be iodine. So why iodine? Well, it's a great oxidizing agent, and it has this amazing visual trick up its sleeve involving starch, which we'll get to in just a bit.
Okay, and this is where it gets really interesting. Using iodine leads us to two powerful methods with, let's be honest, very similar and kind of confusing names: iodimetry and iodometry. They sound almost the same, but you're about to see they work in fundamentally different ways. And this gets right to the heart of it. It's the single most important difference. Think of iodometry as the direct approach, one straight line reaction. Iodometry, on the other hand, is the indirect method. It takes a couple of steps to get there.
So to be clear, both of these methods are built on the same redox chemistry, and they're both used for incredibly precise measurements. The real difference, the thing that sets them apart, is the kind of substance they're designed to measure. You'd use iodimetry for one type and iodimetry for a completely different one. All right, so let's zoom in on that first method, iodimetry. We're going to break down exactly how this direct approach works, step by step. You know, the name itself actually gives us a pretty big clue. In iodimetry, our titrant, that's the solution we're adding from the top, is a standard iodine solution, and it reacts directly with our unknown analyte down in the flask. It's just a single, straightforward reaction. Let's walk through it. First, you take your sample, which has to be what we call a reducing agent, and put it in a flask with a little bit of starch indicator. Then, from this long, skinny glass tube called a burette, you slowly start adding your standard iodine solution. The iodine instantly reacts with your sample until, bam, you see this sudden, sharp color change. That's your signal. That's the endpoint. So let's talk about the roles here. In a redox reaction, you always have one chemical that gives away electrons and another that takes them. In this case, iodine is the oxidizing agent. It's the one that takes electrons. And that's exactly why iodimetry is used to measure a reducing agent, a substance that's ready to give away electrons. See? They're perfect reaction partners. Okay, now for the fun part. Let's talk about that color change. This is the magic moment where we literally see the invisible become visible, and it's the most crucial signal in the whole experiment. Picture this. As you add the iodine drop by drop, it immediately finds a molecule of the analyte to react with. It gets used up instantly, so the solution stays completely clear. Every single drop is consumed as long as there's even a little bit of analyte left. But then, eventually, you reach what's called the equivalence point. This is that precise moment when the very last molecule of the analyte has just finished reacting. For a split second, the flask is perfectly balanced. And this is the critical moment. The very next drop of iodine you add has nothing left to react with. It's now just one single excess drop of free iodine floating around in the solution with nowhere to go. And here's where the magic happens. That one extra drop of iodine finds the starch indicator that's just been hanging out in the solution this whole time, and instantly, the entire flask flashes a deep, unmistakable blue. So what's actually going on here? Well, once the main reaction is over, that free iodine molecule immediately grabs onto the starch, forming a totally new molecule called the starch-iodine complex. And it just so happens that this new complex has a vibrant, deep blue color.
That's your signal. You stop adding titrant, record your volume, and the experiment is done.
Okay, now that we've got a really good handle on iodimetry, let's put it head-to-head with its counterpart, iodometry, just to make the differences crystal clear.
This table really lays it all out for us. You can see how they're almost mirror images of each other. Iodometry is direct, 1 step. Iodimetry is indirect, 2 steps. Iodometry measures reducing agents, while iodimetry is for oxidizing agents. You even use a different titrant. And get this, the endpoint is the exact opposite. With iodometry, you're waiting for that blue color to appear. With iodimetry, you're actually watching for the blue color to disappear. So if there's one thing to remember about iodometry, it's this. It is simple and direct. The iodine you add reacts right away with the stuff you're trying to measure. It's a fantastic one-step process for figuring out how much of a reducing agent you have.
Iodimetry, on the other hand, is the indirect method. In its first step, your analyte, an oxidizing agent, reacts to create iodine. Then, in a second step, you titrate the iodine you just made. It's a little more complex, sure, but that two-step process is what lets us measure a whole different class of chemicals.
And there you have it, a deep dive into iodometry. A simple color change lets us precisely measure something we could never see with our own eyes. It's a perfect example of how chemistry can make the invisible visible. It really makes you wonder, doesn't it? What other secrets can a simple color change like this reveal? Thanks for watching.